Give an example of beneficial random genetic mutations.
A key example of a beneficial random genetic mutation is lactase persistence, which allows some adult humans to digest milk, providing a source of nutrients. Other examples include mutations that confer resistance to diseases like HIV or malaria, increase bone density, or allow for higher altitude adaptation.
What causes lactose intolerance?
Lactose intolerance is caused by insufficient production of the enzyme lactase.
What causes insufficient production of the enzyme lactase, thereby causing lactose intolerance?
Insufficient lactase production is caused by genetic factors, such as a natural decrease in the enzyme's production as a person ages, lactase non-persistence, or, more rarely, by genetic mutations that cause congenital lactase deficiency.
Which genes mutate to produce lactose persistence?
Lactase persistence is caused by mutations in the MCM6 gene, specifically in a regulatory region upstream of the LCT gene.
Which genes mutate to produce lactose intolerance?
Lactose intolerance is primarily caused by genetic factors related to the LCT gene, which provides instructions for the lactase enzyme, and the MCM6 gene, which regulates LCT expression. Mutations in the LCT gene can cause severe congenital lactase deficiency in infants, while variants in the MCM6 gene, near the LCT gene, control lactase non-persistence, the gradual decrease of lactase production that occurs in most humans after childhood.
What caused the genetic variations?
The genetic variations in the MCM6 gene responsible for lactase persistence, the ability to digest milk as an adult, were caused by random mutations. Genetic mutations are the ultimate source of all genetic variation. They can occur in several ways. Errors in DNA replication: when cells divide, their DNA is copied. Mistakes during this process can alter the DNA sequence. This is a common cause of naturally occurring, or spontaneous, mutations. External mutagens: exposure to environmental factors, such as UV radiation or certain chemicals, can also cause damage to DNA that results in mutations. Recombination: the shuffling of genetic material during meiosis, the cell division that produces eggs and sperm, can also create new combinations of alleles.
How do you know it is the result of evolution?
Several lines of evidence demonstrate that the genetic variations for lactase persistence are the result of recent, strong natural selection. This process is a classic example of gene culture coevolution, where a cultural practice, dairying, created the selective pressure for a genetic trait, lactose digestion. The frequency of the allele began to rise significantly in later periods. For example, a 2014 study found that by AD 1200 in medieval Germany, the lactase persistence allele frequency was already as high as modern levels, suggesting its spread occurred rapidly within a roughly 4,000-year window.
Is lactose present in human milk?
Yes, lactose is the main carbohydrate in human milk. The human body is designed to break down this milk sugar during infancy, which is why lactase persistence, lactose tolerance in adulthood, is a relatively recent evolutionary trait.
Why would it be a new trait if humans have been consuming breast milk for 50,000 years or more?
That's an excellent question that gets to the core of why lactase persistence is such a clear example of recent human evolution. The key difference is that the body's ability to produce lactase as an infant is a distinct and ancient trait from the ability to keep producing it as an adult. Post-weaning shutdown: in the vast majority of mammals, the gene that produces lactase is switched off after weaning.
If lactose persistence is the result of selective pressure, then it is not random.
It's an important clarification. The mutation itself was random, but the increase in its frequency in dairy farming populations was not random. but was instead a direct and predictable result of natural selection.
How is the gene that produces lactase switched off after weaning?
The gene that produces lactase is switched off after weaning through a mechanism of genetic regulation. After early childhood, the ancestral version of this regulatory element allows for the gradual accumulation of epigenetic changes, particularly DNA methylation, at the MCM6 site.
So the switching off is not a random process.
You are correct that the switching off of the lactase gene is not a random process. It is a tightly regulated and genetically programmed mechanism.
How did the tightly regulated and genetically programmed mechanism come about?
Switching off mechanism for the lactase gene evolved because it was advantageous for mammals to conserve energy once they were weaned.
So, the programmed mechanism was also a random mutation?
The genetically programmed mechanism for switching off the lactase gene was not a random mutation, it is the ancestral default for mammals. It was a random mutation that overrode this programmed shutdown that led to lactase persistence in some human populations.
The ancestral default was produced by random mutations.
An insightful follow-up question. The ancestral default mechanism for switching off the lactase gene was indeed the product of random mutations, but not in the way you might be thinking. It was the product of many random mutations over millions of years, guided by natural selection toward a specific outcome, metabolic efficiency for most mammals.
So it was built step by random.
That is exactly right, but it's important to understand the nuance. The ancestral default mechanism for switching off the lactase gene was built step by random mutations that were then refined by non-random natural selection over millions of years.
Calculate the probability of multiple random mutations forming a tightly controlled genetic regulatory mechanism.
The probability of the final product arising from scratch is astronomical, but that is not how evolution works. The probability of the ancestral default mechanism arising from scratch via random mutation in a single, coordinated event is, for all practical purposes, 0.
What is the first mutation in the switching off mechanism?
It is impossible to identify the first mutation that created the ancestral lactase shutdown mechanism because this complex genetic program did not arise from a single mutation.
What would be the natural selection for the first mutation?
The ancestral lactase shutdown mechanism is not the result of a single first mutation but a cumulative process spanning millions of years.
So there have been many random mutations for lactase persistence since there are many variations among human populations.
Yes, that is exactly right. The genetic evidence shows that lactase persistence has arisen from several different, independent mutations across human history. This is a clear example of convergent evolution, where different populations facing similar selective pressures arrive at the same functional solution through different genetic means.
What is the probability that the random mutation occurred at the same time as dairying started?
The probability of a specific random mutation, like the one for lactase persistence, occurring at the exact same time as a cultural practice, like dairying, began is exceptionally low.
What is the probability that the random mutation occurred in different populations at the same time that dairying started?
The probability of independent, random mutations for lactase persistence occurring in different populations at the same time as dairying started is extremely low.
How low compared to the first probability?
That's an insightful question that gets to the heart of convergent evolution. The probability of different, independent, and random mutations occurring at the same time in geographically distinct populations is infinitesimally low, effectively 0. However, evolution is not a single, coordinated event.